In my lab, we study signaling proteins that play a vital role in physiological systems like we smell, we breathe, we taste, and there are some other proteins that pathophysiological conditions like cancers, some immune disease, and so on and so forth. And these proteins are GPCRs. We call them G protein coupled receptors. They are a membrane protein, the largest family of membrane protein. They reside on the plasma membrane, bind extracellular ligands outside and signals inside. And these proteins are also called as 7 transmembrane domain proteins because they have 7 transmembranes. And in humans, there are more than 800 GPCRs already characterized. And these to understand the signaling mechanism, we have a receptor in red that is a beta-2-adenosine receptor. When a drug can adrenaline bind outside, it creates dynamics of those receptors, and that's actually activate the receptors inside the heterotrimeric G proteins, and G proteins can go to the different signaling pathways. And human GPCRs are targets for many drugs, and there are some FDA-approved drugs that targets for human GPCRs, and there comes with like inhalers, pills, and also injections, and many of you right now currently using one of these drugs here in this audience. And there are 34% of the FDA-approved drugs targets through GPCRs. There are more than 475 approved drugs already, and that targets 108 different targets in GPCRs. They are like 300 in clinical trials as of now, and around 180 billion US dollars revenue generated by GPCR drugs. Not only that, they are also cause several side effects and addiction, like there's oxytocin here. That's basically is drugs with the pain medicine. That's when it has a severe side effects, and also the addiction, and more than 68,000 opioid involved overdose death in the United States according to NIDA website. And GPCR drugs are characterized based on their efficacy. Some then decrease the efficacy, some increase the efficacy. On the right, you can see there are some ligands that basically binds to adenosine receptors that are more potent to different drugs. And what we are looking for here is we're looking for a GPCR drug that binds to the GPCR, signals to them inside the cell, okay, this is towards a more therapeutic and less side effect and stop the addictions. Can we achieve this one? Yes, we can achieve this because each drug may stabilize a different conformational state of GPCR, resulting in the different efficacy. And I would like to put this quote from the physicist and the Nobel laureate John Feynman. He takes about all these things happens because of the wiggling and jigglings of these atoms. So what we are looking for is basically we are looking for the dancing of these GPCRs when the ligand binds, and then how they interact inside the cell. So GPCRs, they are in the two states, in active and active states. When you go from an active state, there are those helices, they move in different orientations shown by the red arrows there. However, the spontaneous transitions from the active state to inactive state is still unknown. So is it possible that we can study those? Yes, it is possible at the molecular level. That's what in my lab, what we do is we use the single molecule fluorescence, which basically allow us in nanoscale detection of individual biomolecular complexes and their dynamics. On top of that, they also give the transient intermediates, which are not actually seen in ensemble experiment. And I would like to put this one like looking for Alabama fan when the ball's playing at Neyland Stadium, because we have to zoom the camera to see the Alabama fans there. So same way we look at the molecules. We look at the molecules, and then we go and zoom each molecule and look at their dynamics and their informations. So how we do that one? So we grow the cell, we ask the cell, okay, just go and produce the protein. We harvest the cell, and then we make a membrane fractions. Out of membrane fraction, we take out the particular protein of interest, like the GPCRs here, and purify them using a nickel affinity chromatography. After that, the pure protein, we put that into the nanodiscs. The nanodiscs is kind of a native-like environment of membrane and the membrane scaffold, lipids and membrane scaffold proteins, and we label them with the CY3 fluorophore for our visualization. information immobilized on the microscope slide using biotin and streptavidin interactions. Now this slide is ready to look at the microscope. When we put that on the microscope, which is on the left side, on the right side, I have a schematic diagram of customized single molecule total internal reflection fluorescence microscope in my lab. That where we can see that one emission from the molecules. We collect them and capture on the CCD camera like a beautiful twinkling little star, like something like that. So we look at those molecules and focus on each of those molecules, and we see that some molecules, they fluctuate into 2 different intensity states. Otherwise, on the other hand, some molecules, they fluctuate into the 3 different intensity states. So basically, we can look at the heterogeneity of the molecules, looking at one molecule at a time, and then from this photobleaching, which is shown by the arrow, shows us that we are looking at only one molecule, and then some ligands, they don't make any changes on the distribution. We can see still 2 for GM 2, 4, 1, 3, 8, 5, but the NICA shows that 3 distributions. That means each drugs has a different efficacy for GPCRs. And from that information, when we look at the careful look at these transitions, what we saw is like going from state 1 to state 3 that always go through the state 2. So there is the state 2 is a transient intermediate state, which is obligatory for GPCR activations, which are not captured from any crystal or any cryo-EM structures. So what we learn here, like we learn that single molecule, yes, single molecule fluorescence can be used to screen GPCR-based drugs, and we can separate the drugs based on the therapeutic or the addictive or the less side effect by using this kind of technique. And that means I call that as a one drug at a time. And with that, I would like to thank my students, my undergraduate students, postdocs, and the National Institute for Health for Money. And thank you.